The overall goal of this set of procedures is to immunize mice with radiation-attenuated Plasmodium parasites to facilitate the analysis of myeloid cell recruitment in the mouse skin and draining lymph nodes.
This method can help answer key questions in the malaria field, such as what are the cellular actors involved in the innate immune response to live attenuated parasites?
Between 18 to 25 days after the infectious blood meal, collect and cold anesthetize infected mosquitoes in a 15 milliliter tube on ice. Next, carefully invert the tube onto a petri dish on ice and expose the insects to a 12 kilorad dose of gamma or X irradiation. Collect the irradiated mosquito salivary glands in 15 microliters of DPBS on ice. And then, gently crush the glands to release the sporozoites.
The injection of a highly concentrated parasite suspension in a small volume is critical, so it is important to harvest a sufficient number of salivary glands from well-infected mosquitoes, as evidenced by their robust expression of green fluorescence.
Resuspend the parasite suspension prior to filtration through a 35 micrometer strainer adapted to a 1.5 milliliter microcentrifuge tube. Using a microscope counting chamber, count the total number of isolated sporozoites and adjust the parasite suspension to an 8.3 times 10 to the 4th sporozoites per microliter concentration in